Good morning, everybody, and welcome to ICTS. I'm very delighted to welcome you all, invited speakers and participants. One of the organizers, we are waiting for him. He's on his way. So Prabal Maiti will join us soon. And Professor Ambarish Kunwar, who was also co-organizer, is here. So in case you have not met him before, he's over there. And I'm Debashish Chowdhury. So we planned this ICTS program before pandemic, but unfortunately because of the pandemic, it got delayed. So I'm glad that now at least we are able to organize. I'm sure the student participants at least that we would have that day and the student participants that we have today, there are very few who are common, but that's the way things are. So without taking much time, I have just a few more things to say. First thing is that about this institute, many people would like to know, and Center Director, Professor Rajesh Gopakumar, will tell you about this institute in brief introduction before Professor William Goddard's lecture in the afternoon today at 4 p.m. And one more thing that I have been asked to tell you is that if you have any question, then please use the microphone. The lecture is getting recorded. So please use the microphone so that it is audible to others also, as well as to the speaker. So let's now move to the actual program. First speaker, of course, is Professor Heiko Rieger, and next will be Professor Masako Sasai. And in the first session, Professor Professor Raja is, he has kindly agreed to chair this session. So, Professor Raja Paul.
Good morning, everyone. So it's a great privilege for me to introduce the speakers in the first session. So this is after a long time, we are back in ICTS in the physical mode. So it's exciting, I believe, for everyone. And this morning we have two talks. Professor Rieger and Professor Masaki Sasai would be giving two lectures. So first talk is given by Professor Rieger, who is he's a faculty in the Department of Theoretical Physics in Saarland University, Germany. Professor Rieger's, by training, he's a physicist, and probably if you are, he has a phenomenal contribution in the beginning of his career in the field of disordered systems, especially in the spin glass systems. And but later on, he sort of started working a lot in the area which overlaps with the biology and interactive systems, and especially in the area of self-organization, collective behavior, and many other things. So this morning, he's giving three lectures. So in the first lecture, he's going to share his views on diffusion, first passage processes and reactions. So thank you, Professor Rieger.
Thank you very much, Raja, for the introduction, and Debashish and the organizers for giving me the opportunity to lecture here. It's a privilege, as Raja said, to open this this program, but also a burden, because I'm pretty unsure which format I should present. I have three lectures, each lecture 1.5 hours, so it cannot be a normal talk. But on the other hand, Debashish also told me that I should put some sort of my own stuff into the presentation. So I decided, I set the stage. Nobody is before me. I'm the first one, so I try it. Do a mixture between presentation and Blackboard. And you can actually vote what you want, because I have also a lot of presentation prepared here. Even 1.5 hour long. So if you don't like my Blackboard presentation, just tell me, then I will switch to slides with movies and so on and so on. So since I start this this program, I thought by myself I can set a little bit the stage. The name of the program is Statistical Biological Physics. I'm from education a statistical physicist, so this I understand, from single molecule to cell. Already here, I was a bit confused. So should I only talk about single cells or also about many cells and so on and so on? So we will see. The nice thing about statistical physics and physics in general is simply that many of the concepts and ideas work microscopically as well as macroscopically. So we will see. The focus, according to the program description, is fluctuation and noise in stochastic biological processes. And to make that more concrete, so that when you consider stochastic processes in living cells, the idea there, the major challenge is essentially for us and also for the biologists, you have a highly noisy environment, and nevertheless, it must be guaranteed, must guarantee the operation of the molecular components of the cell. So there are lots of questions that should be addressed with statistical biological physics. Essentially, what are the specific sources of noise in the cell? How can the cell be robust to noise and attenuate noise effects? Major problem is you have a stochastic genotype. So there's a stochastic gene expression, but you always get, not always, but most of the time you get a reliable phenotype. You have a noisy and crowded environment. So what about diffusive transport? Does that work at all? On the level of molecular machines, how does the conversion of chemical energy to work operates? What are the physical limits of biochemical signaling? Sensitivity of biochemical sensors. The other question is, can you turn it around? Can a cell actually exploit noise to enhance its performance or survival of the host organism? That sounds mysterious, but when you think about stochastic gene expression, then it might be favorable to produce a phenotype which is not always optimal, adapted to the current environment. It might be superior when the environment changes. That is what it is meant. Yeah, and what is the role of self-organization in the formation and maintenance of subcellular structures? The most prominent of which is certainly the cytoskeleton, which you will certainly hear of. So potential topics would be diffusion in cells, stochastic ion channels, polymers in molecular motors, topic of sensing the environment, yes, chemotaxis, mechanotransduction, stochastic gene expression. I mentioned already regulatory networks. I won't mention, but I'm sure that will come in the program. Transport processes in cells, very important. Anomalous diffusion, the narrow escape problem. Sounds technical, but it's simply the problem that has to be solved by cell in daily operation, simply to transport or to bring some molecule or organelle to specific location, for instance, on the membrane. And that takes time, and how much time is essentially answered by mathematics context of narrow escape problems. Nanopores and channels, try to put particles through a nanopore or even whole polymers, exclusion processes, when you have sort of single file transport through a long nanopore, random intermittent search process. Topic could be self-organization in cells, even 2 times active processes, cellular length regulation. There will be a talk about cellular length regulation, cell mitosis. There will be a talk about that too. cell motility, this whole area, fascinating area of understanding mechanically cell migration. The other part of self-organization is connected to reaction diffusion models and the emergence of Turing patterns, min protein oscillations is a big, it's been a big problem, and cell polarization. Okay, of course I will not cover that all. It's sort of, these are the potential topics of this program. So I will talk about essentially, the first part about diffusion, the second part about first passage process, and then in the third part, I wanted to talk about reactions, but then I decided maybe I switch to something different, more shiny, let's say. I want to talk about biophysics of killing. This is essentially a topic, subtopic of immunobiophysics. So essentially, try to understand how T cells and natural killer cells perform killing of target cells. And that is the third lecture. So that will be tomorrow evening or late afternoon. So we can make a poll. You tell me whether you would like to have this one here. I will talk about the status of ligand receptor binding. So not the one that you usually see in chemistry textbooks, but understanding of ligand binding from the point of view of statistical physicists. Then the MWC model for an allosteric receptor, for instance, or not kinetic proofreading, kinetic proofreading during protein synthesis, T cell activation, and then finally the Gillespie algorithm. So we will decide tomorrow. But the first lecture will be about diffusion, and the second about first passage time processes. So I will now sort of diffusion go slow pace on the blackboard. So essentially, I'm sure you know that part at least. When you have a diffusing object, then there is a linear relation between the mean square displacement and the time that is observed. That is certainly already sure that you know. That is here sort of the mean square displacement of an object that is moving randomly through space. That is the time interval measured. This is the diffusion constant, dimension length square over time. And 2 times d, d is the dimension. In one, it's a d. In one dimension, it's 2. In 2, it's 4. In 3, it's 6. That you know. Relevant for, it might sound mathematically, but actually it's relevant. For instance, when you look at single cells, the cell interior, that is 3-dimensional diffusion, or membranes. Okay. And in 1D, it's axons. All 3 cases have been covered by single particle tracking quite a lot. So when you look at these sizes or length scales and time scales in bacteria, the volume is roughly 1 cubic micrometer, like Escherichia coli, for instance. The length is 1 micrometer. So for is that serious that I should not cross here? Okay. Yeah, but I don't have periodic boundary conditions, so So essentially, I need to, okay. Still bigger, okay. Okay, start again. So bacteria, V roughly 1 cubic micrometer, length scale roughly 1 micrometer. So when you have a small particle, small molecule, or then the diffusion constant is typically of the order of 100 or even, in the case of calcium, for instance, that's what I mean with a small particle, it's 220 micrometers square per second. So that is what is meant. For a larger particle, protein, a protein, for instance, I'm too small again. Oh, I see. Yeah. D is roughly 10 micrometer per second. So that means for a small particle, for a bacterium, it can traverse by random diffusion the whole bacterium in 1 microsecond. Here, it takes only 10 microseconds. For yeast, the volume is already 30 micrometer cube, length, let's say 3, then the time scale is already of the order of fractions of a second for a small molecule and 0.1 second for a protein. Now, a mammalian cell, like for instance, HeLa, cell line, then you have all V roughly 3,000 cubic meter, L roughly 10 micrometer, and tau is then roughly 1 second for diffusion, and for small protein, for small molecule, and 10 seconds. So you see already what the limitations of diffusion are. And when you talk about cell-cell communication or even organ-organ communication, it's clear that other transport mechanisms have to come into play, which you know, of course. And of course, when you have a large cell and you have to put large cargo from one place to the other, then there is no way other than active transport. With molecular motors along cytoskeleton filaments. Just an example. D is so that is fast on microscopic length scales, in micrometer and milliseconds, and slow, still slow on macroscopic length scales. So when you have, I just mentioned, when you have an axon, which is of, I think it's Xenopus, up to a meter long, 1 meter. With diffusion, that is that needs 10 to the 9 seconds. So years. It's clear that signaling along axons has to work in a different way, and most probably you know that all. Okay. The mathematical description of diffusion, I briefly summarize. There are many ways. Essentially, The first one I mentioned here is a stochastic differential equation. Second one is the Fokker-Planck equation, and third one is master equation, essentially. So, description. Longeval, that is what you usually see in textbook. Time derivative of the coordinate or position of a particle is, now usually in math books you find a coefficient A here and a coefficient B for the noise. For physicists I make it more concrete. So you have here a force acting on a particle, yeah, and the friction coefficient gamma plus square root of 2d times psi of t. Yeah, where psi of t is Gaussian noise with the two, for instance, 0, 2 moments like this. It's delta correlated noise. Usually it works for these kind of coefficients. As soon as you have, for instance, the coefficients that depend on space, it's better to do it mathematically, with the Ito or Stratonovich stochastic differential equation. Well, as a physicist, it's easy to understand. You just write this here as a differential, yeah, and then multiply this with this dt, and then you get, here this is dx over dt, then you get essentially this one here. It's the same, gamma is the drag coefficient. And F force. Experiment with the colloid in single particle in oil, for instance, it's in a gravitational field, so a constant force. So then the velocity, you can define F divided by gamma as the velocity, and obviously in the solution of this differential equation is simply square root of 2d integral plus d times t, which gives you the famous formula for the mean square displacement, this in one dimension. So a stochastic process described by the Langevin equation or Ito stochastic differential equation is actually a Markovian stochastic process. It means essentially the transitions from one state to the next do only depend on the current state of the system and not on the past. So the process doesn't have memory. Oops. And for Markovian processes, I mean, there is a whole machinery that can be used and immediately leads to the other two concepts that I mentioned to describe. Where is here the border? Here's no border. Okay, maybe here, huh? So note x of t described by 1 or 2, with 1 Celangeron equation, 2 is Markovian. So it's a Markovian stochastic process. So it is fully described by P, X, and T. So the transition probabilities, there's a probability to find the particle in state X and T when it was at time T0 in state X naught. For a Markovian process, one can so Markovian follows these so-called Chapman-Kolmogorov equation, essentially, which is nothing more than mathematical expression of the fact that when you go from xt to x naught, t naught, the probability from here to here is the probability to go from here to an intermediate state and then further integrated overall. So means P of x and t, x naught, t naught is equal to integral minus infinity plus infinity dx prime pxt x prime t prime p x prime t prime x naught t naught. And this is what you have for Markovian processes. Now, since for almost all processes or many processes that you consider, a kind of locality holds. That means if there is a transition, for instance, in space, this transition is only to nearby places. In jumps far away are exponentially suppressed. That means, and then with this locality principle, essentially you can do a so-called an expansion of this equation for essentially infinitesimally distance between t0 and t. This is the so-called Kramers-Moyal expansion. And when you have, maybe I show it Kramers-Moyal expansion, then plus locality gives you the Fokker-Planck equation. D, dt, p, x, and t, a drop. I condition this probability already to the initial condition that at the beginning, at t0, the particle was at x naught. Is minus 1 over gamma dx fx px and t plus d times okay. This is essentially when you deal with many problems in connection with diffusion, but also in connection with active transport and in connection with active matter, a central equation. Just a side remark concerning active matter. You can put the gamma here and then note that this here is the velocity. And then you have here the sort of the advective term, so or in connection with active matter, it's the self-propulsion with a velocity v. Here it's externally driven by a field by a force F in the diffusive term. And then in active matter, here is an additional equation for the self-propulsion speed. Yeah, so this is an a central equation not only for diffusion but also for active transport. Few remarks. It's obvious that for d equals 0, let's say F0, for F0 you get the diffusion equation. Just P dot in short notation. And for d equals 0, so the diffusion constant is 0, you get the deterministic Liouville equation. So the solution of this is simply integral minus here's delta function. So this here is a delta function. Yeah, instead of you're keeping the particle on the classical path. Yeah, that is the solution of x dot equals f of x over gamma. And this here is the probability distribution of initial states. Yeah, essentially this is the Liouville equation. Yeah, and indeed, from here you can go away from biology to classical thermodynamics and equilibrium physics. In equilibrium, you would have a temperature T. Sometimes this is also assumed in biological context, yeah, when you discuss diffusion in the cell. There is absolutely no reason to do this, except that you do not have, because there's also active noise, not only thermal noise. But as long as you sort of do not clarify the source of noise in the cell, is the best what you can do. And in equilibrium, we have, if you assume, yeah, how to put it, in equilibrium, in an equilibrium system, the Einstein relation holds. And that is the relation between the diffusion constant and the drag coefficient or viscosity. Essentially saying d times gamma is equal to k Boltzmann T. And in that case, the equilibrium distribution or the stationary solution of the Fokker-Planck equation is simply the Boltzmann distribution. E to the minus k minus u of x over kbt divided by the partition function or times the normalization. Where u essentially, or the gradient of mu is f of x over gamma. Okay, so that is essentially what you have in equilibrium. But let's now forget that already. The Fokker-Planck equation itself can be written as a continuity equation because the term here can be written as a divergence. Yeah, so essentially, the Fokker-Planck equation is nothing more than a conservation law for probability. FP, Fokker-Planck equation, written as equation dp over dt is minus d times minus dj over dx. With probability flux is 1 over gamma. So the advective term p of x and t. No. Yes, du dx. So that is the potential for this force. Where was I? Minus d dp xt over dx. Okay? That is important in many sort of formal aspects, but also clarifies the physical meaning of this equation. Just to give you an estimate, when you have a spherical particle, just concerning this drag coefficient, when you have a spherical particle that is moving in a fluid with some viscosity eta, then the Stokes relation holds. A spherical particle in fluid with viscosity eta, that's the Stokes relation. Gamma is 6 pi times eta times r is the radius of the particle, of the spherical particle. Now, when you have a, say, a nanoparticle, yeah, so r equals 10 to the minus 9 meter, a nanometer, yeah, and viscosity of water is something like 10 to the minus 3 kilogram per meter second. That results in a diffusion coefficient of 100 micrometers squared over second. I think, oh, this is fast. Yeah, nanometer. This is the size of proteins in cells. Yeah, so actually they should diffuse as fast as roughly calcium. No, that's not true because the cytosol is not water. So essentially, a reasonable number that you could insert here for the viscosity of the cytosol is 30 times, or more, even more than the 30 times the viscosity of water. And that then already that also then leads, of course, to 100 over 30 for the diffusion constant. Okay. It is clear everything that I wrote here is for one dimension, of course, and just a little bit of vector notation, and you immediately have this equation in 3D, the divergence of f times p plus d times Laplace of p. And also here, you see immediately that the right-hand side can be written as the divergence of a current. The boundary conditions of the Fokker-Planck equation. We will encounter it a couple of times in connection with first passage times also, therefore I mention it here. Usually, there are two kinds of boundary conditions, at least, and mixtures of that. Yeah, that is the Dirichlet boundary condition, conditions for Fokker-Planck. For instance, when you have sort of prescribe the probability on the boundary so x on the boundary of the domain, that is Dirichlet. When you prescribe the probability flux on the boundary where n is the normal, on the boundary that is g of x and t, that is von Neumann. For instance, when you set f to 0 at the boundary, then you talk about the so-called absorbing boundary conditions. So there is a probability flux going into the boundary. And when you set G to 0, then it is a reflecting boundary conditions. There's no flux going through the boundary. Okay. Yeah.
Assumptions that you have made, there was one more while writing those, that is those equations for the Brownian particle were written for time much greater than gamma inverse, because otherwise there will be drift term essentially for very short time which would decay exponentially, and that is also related when d is equal to 0, then Einstein relation would imply that gamma is infinite at a given temperature, and that is instantaneous relaxation, and that is because that term is not there, otherwise that term would.
Yeah, it's true.
Yeah, so sometimes students they interpret that as if diffusion from the beginning, but the diffusion would not be from the beginning at this up to of the order of 1 by gamma. Right?
I think that one should have always in mind that diffusion is not a natural pattern of movement of particles in nature. It's a mathematical description of something. Yeah. So how the particle actually moves, I mean, it's much more realistic to assume a molecular dynamics simulation of it. Yeah. But in essence, when you even look at the molecular dynamics simulation of a particle, a tagged particle, for instance, in the fluid, with many other particles, yeah, then when you have this very detailed mathematical description, and even can put it on a computer and simulate it. But when you look at your data from far away, then you will see that, oh, I can describe the movement of this tagged particle with a diffusion equation. But this is a description of a larger scale, as Debashish said. Yeah. And when you sort of turn on your microscope, then you will you will see that on a microscopic length scale, it's not diffusive motion. Yeah, it's a ballistic sort of between collisions with environmental particles, and even also. Yeah. Okay. There are two other remarks I wanted to make, but I will skip them. One is the sort of on the Ornstein-Uhlenbeck process, which is sort of one where the for the essentially an equation with an exponentially decaying correlation function. I skip it. Then a remark about when you write down the stochastic differential equation, then you see, okay, in order to handle this, you have to write, to integrate it essentially. And then in the literature, when, in particular in the more mathematically oriented, you immediately, you encounter Ito interpretation and Stratonovich interpretation. It essentially comes from the fact, as long as you deal with non-multiplicative noise, as we did here, where you have just a diffusion constant in front of the differential of the stochastic. Here. Here, have just a constant. You do not have to bother or here with Ito or Stratonovich. But as soon as the noise depends on space, then when you sort of integrate this formally and write the integral as a Riemann sum, then you will encounter that you have a product of this term here, which is now dependent on space, and this term, which is a random variable. And when you write the discretized sum for the integral, then you will see that there is a correlation between infinitesimal, but there is, and this term here, which depends on space, and this term here, which for each space point is a random variable. And depending on how you interpret this term, that is then the Ito interpretation or the Stratonovich interpretation. I don't think that I will deal with multiplicative noise, but I just mention it here. In particular, we will essentially deal only with problems where you have a constant here. So I just write the remark, multiplicative noise, So when dx of t is, let's see, I forgot here, dt, sorry. Dx of t is a function ax dt plus a function bx of then d omega of t. Then this here, the x dependence, then you should bother with Ito or Stratonovich interpretation. Even the Fokker-Planck equation in both cases for the interpretation of this, what that actually means when you integrate it, is different for Fokker-Planck or for Ito. It's a difference between a diffusion equation, let's say, Stratonovich, with space-dependent diffusion constant, for instance. That is Ito. Because space-dependent diffusion constant means that this factor is space-dependent, and P dot equals d of x times gradient d of x of P. This is Stratonovich. And oops, I'm sorry. This is Stratonovich, sort of where you have the d of x inside. So depending on, there is no truth. These are both mathematical descriptions which are essentially equivalent. And when it comes to macroscopic variables, hopefully what comes out is at least transferable to each other. But I just mentioned it to you. Already when you have, for instance, space-dependent diffusion constant, you can decide for this equation or for this equation. Okay. Now, after these formalities, I have now the choice of zillions of possible applications. And what I will do is I tell you a little bit about the so-called Smoluchowski reaction rate. And with that then about the phenomenological description of the physics of chemoreception. Should I equilibrium just Planck equation as a continuum equation? This we can. I will talk, first passage comes tomorrow. Should I derive it? Okay. Yeah, since you mentioned it, it sounds mysterious. Maybe I already make a remark here. There are two equivalent descriptions, even on the level of partial differential equations for a process. One is the Fokker-Planck equation here, which goes this way. The other way is the backward Fokker-Planck equation. Essentially, the Fokker-Planck equation is an equation for the progression of probability. Essentially, you compare probabilities sort of in the future. And the backward Fokker-Planck equations is the equation where you compare infinitesimally distant initial conditions. It looks pretty similar. In particular, when you have a constant term here. But the signs are different and the interpretation is different, and the backward equation actually it's necessary, or not necessary, it's convenient to derive equations for first passage times. But I'll tell you about that tomorrow. The applications. So I won't do big calculations here, but essentially show you just the way the Smoluchowski reaction rate. It is essentially when you have diffusing particles that can essentially react with one another, then there are essentially the rate of reactions is limited by two processes. One, the first process is diffusion. Of course, they must encounter first somehow. And the second is then essentially the reaction itself, the chemical reaction itself. That can be super slow, so it's an inert system, or it can be super fast, like for instance, infinitely fast. Then the rate limiting process is essentially diffusion. So when you assume that limit can be considered and is considered here, you can assume that the particles, once they encounter, they immediately react. So you ask, okay, nevertheless, when you distribute them over the medium, they do not immediately react, cannot, because they are not close enough. They have first to encounter each other, to diffuse each other to each other. For diffusion limited consider A plus B gives you a compound AB. Assume A and B react immediately upon encounter. then K in is limited by encounter. Right. Via diffusion, of course. Note, when you would write, sort of describe this process on the level of the law of mass action, then you would write such an equation and immediately, naively would insert k equals 0, k equals infinity, because that is what you do on this level of description, which is a mean field level. But here, sort of we think in terms of a reaction diffusion equation. So you have this chemical process plus diffusion. So that means it's not well mixed. The particles have first to encounter and can then react. And that modifies the chemical reaction rate. This is actually a first passage process too, but we can also describe it by consider the probability fluxes essentially. So we take particle A as fixed. Maybe I make it larger. A, in reality, it's much smaller. Yeah? So this is particle A, which has a radius A. In particle B, so this is fixed in space, diffuses around. Yeah? And as soon as it, so this is of course also, it has some size, but I neglect this. It's sort of the effect. Essentially, you can take both. This is radius B, this is radius A, can replace by a system described here by a point, and here radius A plus B. Yeah. Okay, and when it hits here, then essentially, then it immediately reacts. So this is the target domain. Omega, and the boundary is an absorbing boundary. And C of x and t is concentration, B concentration. Concentration of B molecules. Okay, that then can be described by a diffusion equation. This problem is absorbing boundary conditions on delta B, delta omega. And initial condition for time 0, you have a given concentration, and at infinity, so far apart from the reacting part, it levels off to the initial concentration. And what you should calculate J, the flux through the boundary. Calculate flux through boundary. The flux through the boundary is essentially what determines the reaction rate, the Smoluchowski reaction rate. The flux through the boundary is J is equals D times integral omega gradient of C times dF. Okay, now I leave you as an exercise to actually perform this. You see, it looks difficult because here you have vector operators and all that. But this is a radial symmetric problem. And in spherical coordinates can easily transformed into one-dimensional differential equation. And the solution is of the Fokker-Planck equation with the corresponding boundary conditions is C0 times 1 minus A over R plus A times C0 over R error function R minus A square root 4 dt. Now note that this here is vector R, and this is the modulus of the vector of R, just the radial distance. That means the flux is, when you would like to understand how this solution looks like, essentially you have the target here somewhere in this region when you look from the side, let's say, and see then from C0 there drops, essentially. And here essentially you have a slope, that means in this distance R, and this is C. This is A. Yeah. So it goes here from infinity, from C0 to some boundary. You have a slope here, and that gives you then the current, which is 4 pi d 4p A times d 1 plus A over square root of p dt. And that goes for t goes to infinity. So here you have still a time dependence in the flux, but in the stationary limit, it goes to 4 pi A times d times c naught. Oh, I forgot c naught here. Then this here, so the flux is some rate times the concentration, so this is the K. So the Smoluchowski is 4 pi E Yeah, note that the dimension of K is indeed volume per second. Okay. So even if you have sort of super reactive partners, reaction partners, then in real space, there is still a limit to the reaction simply via this diffusion, and this limit is this rate constant. Depends on the diffusion constant. Obviously, the diffusion, when the diffusion constant goes to infinity, then of course the reaction rate also goes to infinity. Now we use this number, this reaction rate, essentially to discuss a little bit the physics of chemoreception. There's a classical paper from 77 I recommend to read. Yes, this is classical biophysics. So essentially, what is the problem? The problem is, for instance, that you have a bacterium which has chemoreceptors on the surface. Receptors on surface of bacterium. And yeah, think about, for instance, Escherichia coli, that performs this famous run and tumble movement. So essentially, it moves forward for some time, and then suddenly it starts to tumble, and then moves in a different direction forward, and then again. And actually, what these people have done, and many others probably also, have measured the frequency of tumbling. Yeah? And what they observed is when they put the bacterium in a nutrient with a gradient of decay, let's say, we're able to apply gradients of with a decay length, of concentration decay length of a centimeter, let's say. Then they observe that the tumbling frequency actually changed. It is the way. It's not that the bacterium essentially feels the gradient and then steers with its cilia into the direction, like the sailor in the sea, into the direction of the gradient. No, it cannot do this. So what is actually done is the tumbling frequency. So you observe longer straight runs in a steep gradient. And when you have no gradient, anyway, this is the way in which stochastically the bacterium finds its way, finds its sort of moves more probably into the direction of the gradient, so against the gradient. And the question is, how does it feel the surrounding? And the first question in a noisy environment is simply what has to be, how can essentially the bacterium compute or measure the concentration and even concentration gradients of the nutrient. So what the bacterium essentially, the basic hypothesis, is integrates a signal coming from the receptors for this nutrient over time tau average. And for simplicity, assume that the receptors are perfect absorbers. That is where the Smoluchowski reaction rate comes into play. For simplicity, assume perfect absorbers. That means the number of signaling molecules N is proportional to A times D times C times tau average. When I mean signaling molecule is molecules that signal. That means molecules that that are bound to ligand. And the number of those receptors which have a bound ligand is proportional to this time, and to these factors A times D times C. That is essentially the reaction rate, A times D, yeah, times the concentration. This here is a geometric factor, comes from the fact that we have assumed a sphere, yeah, for the absorbing particles. So this will be, the whole computation will be a back on the envelope computation or fly-by-night physics. So I leave away sort of the prefactors and just argue about orders of magnitudes. Yeah, sort of can insert a 4 pi here, but doesn't change the argument. Smoluchowski. And then, I mean, you will find in the paper, I guess, a more involved computation. Here I do, for instance, now what we have to investigate, sort of what is the size of the fluctuations, because it's a stochastic process, yeah, this binding to a fluctuating environment. So how large are the fluctuations in comparison, in concentration, in comparison to the variation of the concentration in a gradient? And when the fluctuations are larger than the difference, the spatial differences that you get in the concentration, when you have a gradient, it's hopeless. The bacterium can't do anything. So let's see. So in order to estimate the fluctuations of this number, the number of bound molecules, just assume the law of large numbers, number fluctuations is square root of, so the relative. So the mean square deviation is proportional to the square root of n. If it everything would be Gaussian, and relative it's 1 over n. That means 1 over the square root of a d c tau average. As I said, in the classical paper, you find a more complete computation, but not very surprising. The same orders of magnitude come out. From the number of signaling molecules, the cell estimates the ligand concentration simply so from n. So the number of bound ligands, the cell estimates C. How can that be? Obviously, there is this relation in equilibrium, this relation dissociation constant in equilibrium. Yeah, just this from the mass X. So C, the ligand, reacts with the receptor. Number of receptors or receptor concentration is then given by this, where this is the dissociation constant. It means when you invert this, that from this you can estimate the number, from a number of bound ligands, you can estimate the corresponding concentration like this. So it means you have small concentrations, small numbers, then you have this proportionality, and that was 1 over the square root. So that is the estimate of the concentration fluctuations that the cell actually would obtain. Yeah?
Yeah, so I meant that N should depend also on the motility of the bacteria, right? Where does that come into this?
Nowhere.
Doesn't
Oh, of course. I mean, the number, this, I don't know whether this is really, I mean, it depends on many things, but whether the number of bound or bound receptors depends on the speed with which the bacteria moves, I don't think, because it.
Then how does it sense the concentration gradient then? How does it know that?
Yeah, I come to that. Yeah, this is the exciting part, actually. So essentially, what the bacterium does is not, it doesn't sense the gradient immediately. It essentially it compares sort of the concentration, yeah, at the beginning and the end sort of the so after tau average, so which is chemically set somehow. So now assume an experiment that we apply a gradient in C, and C varies over a distance delta L in a gradient. It means C roughly like e to the minus L over lambda. So that means in the gradient, you find a concentration variation of the order of delta L. Yeah, and lambda is the decay length of the gradient. And so the condition for reliable signaling would be delta C over C smaller than delta C over C. Now let's make an estimate. A, this is the size of the bacterium. I know that Escherichia coli looks different from a sphere, but let's just estimate length. D. Now this is a fast molecule, let's say. A concentration, we assume a concentration of the nutrient, or the like a millimolar. Millimolar are roughly 10 to the 5 over cubic meter. I did a lot of calcium dynamics with experimentalists, and I had to compute all the time molar into cubic micrometer. You know that one molar is Avogadro's constant per liter. So 10 to the 6 times 10 to the 23 molecules per liter. A liter is, what was it, 10 to the 15? So I always forget the number. So you have to compute the liter, which is 10 to the 3, 10 to the minus 3 cubic meter, into cubic micrometer, but instead of doing this exercise all the time, could then also simply learn by heart that 1 micromolar is roughly 600 molecules per micrometer. So 1 micromolar is, this 6 comes from Avogadro's number, so you can make it even 600 times molecules per cubic meter. So that should then, so a millimolar 6 times 10 to the 5. And let's say, and this is the time scale of turning for the tumbling frequency that is observed in the experiment. Also, tau average is of the order of 1 second. When you insert all this, and lambda, the gradient that is applied by the experimentalists, is 10 to 1 centimeter, so it's 10 to the 4 micrometer. That means delta over C is roughly 10 to the minus 4, which is smaller. That has to be smaller than 10 to the minus 4 times delta L. So that means you're confused. So the 1 10 to the minus delta, the delta C over C comes from this here. Yeah, this is 10 to the minus 4. Yeah. And the other 10 to the minus 4 comes from the centimeter. Yeah? 10 to the minus 4 micrometer, yeah, from the lambda. So in other words, delta equals 1 micrometer. So over the length of the bacterium, yeah, that is too small. Because then the fluctuations by noise are of the same order of magnitude as the difference in concentration that the bacterium would be able to measure between beginning and ending of the average time. But the bacterium moves, yeah, with roughly 10 micrometer per second. And that means, yeah, it integrates then not the gradient over its own length, but it integrates sort of the difference in concentration, yeah, over beginning of its movement to the end. So delta, and that in 1 second, the 10 micrometer per second give you 10 micrometers. And that can then, during movement with V equals 10 micrometer per second. And that should work. Okay, now Raja is standing there. I have still lots of materials, but don't panic. I will stop here. What I still wanted to talk about is anomalous diffusion. Yeah. A few facts, and then about the run and tumble particle, which would have been a particular honor for me to present here because there is a fantastic paper of 2018 from, I think, 5 or 6 people here from Bangalore. And with the run and tumble particle as a model for a bacterium, yeah, like Escherichia coli, but it's also a model for active particles. Yeah? That would have brought me smoothly to my slides, which show my own work about active Brownian particles. But I think I should definitely stop here in order to give the next speaker the opportunity to start in time. Thank you very much for your attention.
Thank you, Heiko, for this wonderful presentation. So we can take a couple of questions. Is that their time? I guess 5 minutes or so. Very quick questions from the students, particularly. Yes.
So we've been very interested in diffusion of different proteins from outside the cell through the membrane into the cell. And a collaborator, Jim Heath at Seattle, found very interesting effects where chirality played a role of maybe 2 to 5 times in terms of the diffusion. So then we have 3-dimensional diffusion to the cell surface. I guess we have 2-dimensional diffusion along the cell surface, maybe 1-dimensional diffusion through the membrane, maybe then 2-dimensional on the inside of the membrane back to 3 dimensions. So practical problems have all these different factors going on to it. We think in the case I just mentioned, it's actually the 1-dimensional diffusion through the membrane that affects the chirality effects, but that remains to be shown.
How should that work?
Well, so it happens, so the case we're looking at is, so it probably has about 100 amino acids, and then one part of it's chiral. It turns out that the chirality, so in the membrane, it's hydrophobic. So then you tend to bury hydrophilic interactions. And it looks like the good diffuser buries the hydrophilic interactions. It does not interact with the cholesterol. The bad diffuser doesn't do it so well. It has polar groups interact with the cholesterol, and that slows it down, we think. So it's very practical, all these issues.
Yep.
Thank you for this wonderful comments. There are other questions from anyone? Okay, if not, let's thank the speaker. And we meet here again at 11 o'clock. Thanks.